In this video, I'm going to explain how to handle data collected with an enhanced gas switching system set up in stop flow and using 3 of the 4 channels that were available. First channel, channel 1, is always set for reference, so that's the background gas. And we didn't have any chamber attached to channel 1. It was just an empty solenoid valve assembly that is connected to sort of run in a loop. And for channels 2 and 3, we had 2 small insects in each chamber, as it was shown in the previous video that explained how to set up stop flow experiments. We used a variable volume chamber that was set to about 8 milliliters. So here we're looking at page 1 of this experimental file. This is where all the raw data is displayed. We've got the graphs of CO2 and O2, the flow of the through the chambers, temperature, and the absolute pressure at the site of the oxygen sensor. We were doing these measurements around 135 mils per minute, running through the chambers with the insects. The background gas was delivered from a gas bag at around 481 ppms of CO2. And here are the user parameters that have to be set up, some of them before the start of the experiment, others can be done after the start of the experiment. The key parameters to set up before the start is, of course, the number of channels. This is a 4-channel system, and you can use less than 4 channels. We had 3 with the insects in channel 2 and channel 3. Dwell time is another parameter that has to be set up before the start of the experiment. Dwell time is the time that each chamber, each channel is being measured. And while that chamber or channel is being measured, the other channels are in the stop flow mode, so no gas is flowing through it. The background gas just vents directly out through the vents of the gas switcher. And the third parameter that has to be set is the mode. Mode in this case was set to 0. In mode 0, reference channel 1, is measured at the beginning of the cycle, and then once all the chambers are measured. So basically you measure channel 1, which is your reference, then channel 2, 3, 4, and then back to channel 1 reference. There is another mode called mode 1, and you can adjust it here by clicking on that user parameter. In mode 1, reference is measured after each channel. So you measure channel 1, which is your reference, and then 2, back to 1, 3, back to 1, 4, and so on. This is a mode that's used when you're dealing with less stable background levels of gases, for example, or for some other reason you need to monitor your background levels more frequently. The disadvantage of mode 1 is that it takes longer to complete your experiment, and there's more time in between measurements of your chambers, and therefore your stoppage time is going to be longer. So this is something that one needs to keep in mind when setting these parameters. I should also mention the manual channel before the start of the experiment, that should be set at 1. So you're starting at 1. This parameter is only used while you're setting up your system and trying to figure out ideal flow rates. You're setting your flow rate with the flow meters, the rotometers, and then you're measuring it here with the flow monitor QG266. So it's important to do that before you start your experiment, and that was explained in an earlier video. But when you start your data collection, this should be set back to 1. Okay, so I will now explain some of this data and some of the symbols on these data graphs. So this is looking at our CO2. You will see these flag symbols on the bottom, and they indicate basically when the different channels are being switched by the gas switcher, and that is important for the software to do the calculations and sort the data. The red cross indicates when the reference channels is switched. So the data that's measured between this red cross and here is your reference channel data. And here again, and again here, this is the reference channel measurements. You see that there's a little bit of a period of time where you're measuring what's left in the lines coming from the previous channel, so that data needs to be always ignored. The red dots represent when sample channels are being switched. So in this case, this is channel 2 is being switched. So data, so measurements between the red dot and this red cross represents data measured from channel 2. And then we have, because we only had 3 channels, channel 3 is actually the end of our cycle, and the end of the cycle is marked by this orange triangle. So in this case, this is the switch from channel 3 back to reference. So the data collected between the orange triangle and the red dot represents channel 3, or the last channel in this system. So one cycle is between the 2 triangles. So we've collected data here from basically 3 full cycles, and I think this went a little bit longer, 25 here. Yeah, so we have 2 full cycles of data. And as I mentioned before, it's important to collect this sort of trial data at the beginning before you start doing your experiments, and that's to figure out the ideal flow rate through your system, the dwell time, and the mode that should be applied. When you look at your collected data, it's important that your CO2 peaks are nice and sharp, that they do not reach 2,000 parts per million, because 2,000 parts per million is the maximum range of the CO2 analyzer. So if you have peaks that are higher than that, you will not be able to measure them. It's important that your dwell time is long enough to flush the peak completely and reach nice steady state before you switch to the next channel. That's very important. It's important that your stoppage time, and therefore dwell time as well, because in this case we have 3 channels that are being measured with dwell time of 2 minutes. That means 6 minutes in between each channel being measured, 6 minutes between the same channel being measured again. So that means it's 6 minutes of stoppage. And that's important to determine whether you're getting sufficient differentials of oxygen. You can go to page 2 here to have a look at your differentials, and you can see that we're dealing with 200, 300 parts per million of oxygen peaks, which is sufficient. The higher the peaks in oxygen, the better. The oxygen analyzer resolution is lower than that of CO2 analyzer. The oxygen analyzer resolution is about 40 to 80 parts per million. So you need to have those peaks, well above, 150, 200 parts per million for them to be measurable. CO2 is not an issue. The resolution of CO2 analyzer is only 1 ppm, so you can measure very low levels of CO2. But all of that needs to be kept in mind when you are setting up all of these initial parameters for your experiment, to ensure that you have that you collect data that's going to be valuable. The other thing to mention here is this solid dark line here represents actually the background CO2 levels, background oxygen levels, and it is calculated from the reference measurements which are done between the triangle and the red cross here. So the reference is being measured here, and it's interpolated between or during the time that the samples are being measured. It's interpolated from those 2 measurements that are done on either side of the samples. And it's this reference that's subtracted from the values, the measured CO2 and O2 values from the samples, and plotted on page 2 in the differentials of CO2 and O2. One more thing to mention here is that the oxygen analyzer, the QS102 oxygen analyzer in the Q Box package here, it measures oxygen in kilopascals, and it also has a pressure sensor that measures absolute pressure at the sensor in kilopascals. And then within the software, the oxygen signal from kilopascals is corrected for pressure and converted to % oxygen. So what you see plotted here is the corrected oxygen and what's shown in the meter and in tables as well. But the kilopascals of oxygen are measured by the analyzer and are shown in the table as well. Okay, so we can move to page 2, differentials, because this is where the user parameters are that have to be set after this initial data collection was completed. And these are O2 delay, delay low, delay high. I'll discuss O2 delay first because this is the parameter that aligns the CO2 and oxygen signals. The CO2 and oxygen signals may not be aligned because the oxygen analyzer comes after the CO2 analyzer. So oxygen is measured after CO2 is measured, and depending on the flow rate, the volume of the chambers, there may be a misalignment in those signals. It is important to align them because they are being used in calculations that the area under these peaks is used in calculations of the volume of CO2 and O2 that's produced and consumed by the animals. So to do that, we need to proceed to a function called examine, and you can obtain this function from either this menu. You can hover over any of these icons to see what they do. Okay, the examine is right here, or you can go to analyze and select examine here. Either way, you're going to get this line appear on your graph, and it's this line that you will use to check whether your signal is aligned. It helps to zoom into your data, so I'm going to do that right now. So just sort of select your maximum and minimum on the x-axis so we can sort of see, you know, one cycle clearly and a couple of peaks clearly. And now when you bring your examine line, you can see that the oxygen dip is about 1 point behind the CO2. So we need to go to O2 delay, and as with any of the user parameters, you can either double click on it and enter the value directly, or you can use these arrows to go up and down to make changes. So I've adjusted O2 delay to 1, and now you can see my peaks are aligned. So once the O2 delay has been set up, then the next parameter to set up is going to be event time. Event time is the parameter that's used in the software to average the data before the switch to the next channel. So it's a time parameter that's set up from the end of that dwell time towards the beginning. And this time period is set up, it usually represents about a quarter of the dwell time, and it's set up, and you have to ensure that it falls on the steady state of the CO2 and O2 signal after the peak has already flashed. So so in this case here, you highlight that area, and then you can go to delta X here to see what that time is, and it's 0.44 of a minute that I've selected here. And you can go here to event time and type in that number, 0.44, enter, and that's your event time setup. The next parameter to set up, two parameters, are the delay low and delay high. These are flags which inform the software where to stop and start the integration of the peak, because that's how the metabolic parameters VCO2 and VO2 are calculated. And we'll go first through delay low. Delay low parameter, you use your mouse and you start to sweep the data from the point where the switch occurred to the point where the CO2 and O2 peaks are just starting to come up. So oxygen seems to be starting a little bit sooner, so this is probably where I would set the value of delay low. And again, you go down here to look at your delta X, and it's set to 0.728. So you double click on that parameter, 0.278, enter, and you'll notice the black dot, which is this the flag for delay low, moves into position where we've selected the delay low time period to end. The delay high, you do the same way, but now you go to the other side of your peaks, which is on this side, and you know, that's probably right about there. Okay, again, you read your delta X 0.936, and that is the value that you enter here, 0.936, enter, and you'll see the green dot, which is the flag for delay high, now moves into position to select where the peak ends. So the software will now know to integrate the peaks between the black and green dots. So once we have set the delay high and delay low parameters, we can now enter, we can now go to page 3 where the final summary calculations are shown, and the summary final data is shown. So I haven't mentioned this earlier, but I should mention it right here, that go back to net zero for a minute, that with stop flow experiments, the very first cycle, which is the data collected here, should be ignored, because during the first cycle, each channel has a different stoppage time, so the data is not representative. So it's you know, basically the first set of points and the first set of differentials here should be ignored. So when we look at the summary, the first set of points here as well should be ignored. You will see the VCO2, VO2, and RQ data. RQ is the respirometry quotient, which is a ratio of VCO2 to VO2, plotted on the graphs, as well as they are calculated in this table. And to see the data, the summary data, you have to scroll across to the end. The VCO2-1, VO2-1, RQ1, this is our reference channel, so these are not meaningful. Our channel 2 data and channel 3 are the purple and orange, and we had there's no data in channel 4 because we did not set up the system to collect data from channel 4. So to visualize these, you have to sort of scroll down because, as I mentioned earlier, there is a single point that's calculated for every dwell time, for every peak. It's calculated, the formulas are explained here. It's calculated from the volume of CO2 and O2 that's determined, divided by the time, stoppage time. So these numbers are going to show up here in this table. You have to click down to get to individual channels. You may have to expand a little bit these columns. And so now what you can do is you can take this data directly from Logger Pro by going Control-C and copying it into a spreadsheet, Control-V, like this. You have to type in the titles of the column yourself. They do not get translated. So VCO2, VO2, and RQ. So you can do that for, you know, all the data for the individual points. Or what you may do is you can also go to File and Export as, select CSV, and then export the whole data file as a CSV file. So give it a name, stop flow, exported, mistyped, exported, and then you can open that file to visualize the data and ensure that it click convert here. So it takes it from CSV into a spreadsheet. You just open that a little bit more here. You may have to expand your columns to see the titles. In this file, you will find that all the data, all the raw data, CO2, pressure, temperature, oxygen, flow, are exported for the whole experiment, as well as all of the parameters, calculated parameters and control parameters, parameters that are used to control the system, to control the gas switching, to sort the data at the end. Everything comes across. So to get your summary data, you gotta come right to the end where the columns with your data are, which is over here. We've got here the VCO2, VO2, RQ for channel 2, and for channel 3 next to it. And it's again, these you will find individual points for each dwell time show up here as channel 2 data and channel 3 data. So you'll have to take this Excel file and get it organized, delete empty rows to create a spreadsheet, a summary spreadsheet, with the data that you are interested in, for any further statistical analysis or whatever it is that you need to do. So a couple of other things that I want to mention is that Logger Pro, when running these experiments, it's really important that you periodically save your data, because Logger Pro doesn't save data automatically. You have to manually go to File and Save As, or save if you've already named the file while the data is being collected. If you're doing experiments that run over a few days, I highly recommend that you actually save the data every day. You stop the data collection every day, save it, under a name for that day, clear the data, restart the experiment for the next day. And this way you will ensure that you have smaller data files for each day than one big, large file. And if anything goes wrong, there's a power outage or computer goes down, that you don't lose all your data. So saving daily, restarting your experiment is a good idea, and saving frequently while you're running experiments is a good idea.